Physics Bites,
Bites,
a science podcast in English by the students of Notre Dame d'Ascension.
Hello and welcome to a new episode of Physics Bites, the English language physics podcast of the students of Notre Dame d'Ascension here in Paris. This is our fourth season. We're starting it off, and as usual, we're going to start off with our senior students. I have two of them today who are going to be talking to us about biophysics. So, how about you guys introduce yourselves, and then we can get the show going.
Okay, hello, my name is Anastasia. I'm in Terminal, and today we're going to talk about the application of biophysics in medicine and a few principles of biophysics.
Right, in general. Sebastian, do you want to introduce yourself to the audience?
Yes, I'm Sebastian. I am also in Terminal, and I will also with Anastasia talk about biophysics.
Right, and you guys have both been in my class since grade 10. This is a third year together, your final year in lycée.
Yes.
And you guys did a
that's right.
really fun and interesting presentation on biophysics, a topic that is close and dear to my own heart because I did biophysics as part of my studies. And I thought you guys did a really, really good job going over the field in general and giving us a little bit of a taste of how physics and biology mixed together. So how about we start maybe with a basic definition. What is biophysics? Is it more biology? Is it more physics? Is it biology applied to physics? Physics applied to biology? How would you define biophysics? Maybe Anastasia, do you want to start?
First of all, biophysics is, it's a relatively young science that emerged as a distinct field in the mid-20th century. Biophysics applies physics principles to biological systems. It is an interdisciplinary field, but leans towards physics in terms of methods while focusing on biological applications. So I'd say it's more physics.
Okay, so in order to become a biophysicist, you would be a physicist. And you're basically applying physical methods, physical ways of dealing with problems and solving problems, but you're applying it to more biological, biomedical context rather than fundamental physics or space and gravity and all these things. Would that be a good summary?
Yeah, I'd say so.
Okay. So maybe now that we have a more or less clear definition of what it is, why don't we go through a short historical record of how this came about? Who are the main figures historically in biophysics? What did they do? We'll maybe do a really short version of that because you did talk about that in your...
Yes, indeed. So as I said, it emerged in the mid-20th century, but its foundations were laid in the 19th century in Berlin by physiologists like Hermann von Helmholtz and Carl Ludwig. But early biophysics faced a lot of limitation due to underdeveloped technology, which prevented molecular level studies, which is quite an important part of biophysics, studying at molecular level systems. The field developed especially after Erwin Schrödinger's book in 1944 called What is Life? Because this book inspired the discovery of DNA's double helix structure by Watson Crick and Franklin in 1943. And thanks to this discovery, there was a cataclysm of new research and new studies from 1950 to 1970, which made the biophysics advance quite quickly at this period, which contributed to the breakthrough.
One little question. So you mentioned Schrödinger, who is from the 20th century, but you said that the science started in Berlin in the 19th century. Do you have any particular scientists that you're thinking of? Did they do any specific thing? Is there like a starting point that we can consider for the beginning, the birth of biophysics?
Oh, yeah, indeed. It's the one I talked about, Hermann von Helmholtz. I don't know how to pronounce.
Who's a
And I believe
in the history of physics with a lot of work that he did on electromagnet. What did he do that it would be considered like maybe the first bit of research in biophysics? Do you have that in mind?
I believe not, sadly.
That's fine. So after Schrödinger, then you mentioned there was a lot of advancement from the 1950s forward after the Second World War. Want to maybe give us a few
indeed.
bit of history?
Well, first of all, in 1953, there was the discovery of DNA's double helix structure by Watson, Crick, and Franklin in 1953, which was quite an important discovery because, nobody could even, I suppose, imagine that DNA was structured that way.
Right, and we would consider that as biophysics because you're looking at the fundamental biological molecule for life, but there was a lot of physics used notably by Rosalind Franklin to get that structure, right? A lot of spectroscopy, crystallography. Okay.
Yeah.
Where do we go from there? So you talked about a lot of advancements, in particular in medicine.
Yeah, such as medical imaging and treatments like radiation therapy.
Right. So a lot of. So what do you mean by medical imaging?
I mean is about MRI, which is a magnetic resonance imaging. Yeah. So it's a method that helps to detect, diagnose, help to detect tumors or brain injuries or problems that you couldn't see just like this on a person.
Well, you can see within the human body, I guess.
Yeah, that's right.
The specificity of MRI is to give very clear image of different tissue, right? And it is a magnetic process. Okay. What do you have in mind? Other maybe medical imaging techniques which are fundamentally.
I think. I think for that I have other known techniques that. Apply on the nuclear medicine. So the one I'll be talking about is position emission tomography,
right?
Which
we call PET scans, right?
PET scan, yes. Which consists on the ingestion or injection of a molecule called a radiopharmaceutical. The particularity of that molecule is that it is an analog molecule to a biological molecule like glucose, but made radioactive. For instance, the one of the most known is called fluor. Is a fluor 18 isotope of fluor put with a analog to a glucose molecule that acts exactly as a glucose molecule, and therefore you will have that molecule spread all around your body and emit the radiations. So, do you want me to describe?
Putting something kind of radioactive in the body, have it go around, and then you're getting the image from the radioactivity as it is spread around.
Yes, and the whole technique consists if you want to detect a tumor. So basically, tumors are a kind of agglomeration of cells that use much more glucose than normal cells. So what we should get if there is a tumor anywhere in our body is a spike in radioactivity in that zone compared to other zones. Yes.
Okay. So there's a lot of different imaging techniques, I guess.
Yeah.
There's a lot of imaging. All right. So you guys decided to divide your presentation into different domains of physics and played a role term like fluid mechanics and all these things. Do you want to give maybe a short summary of each, and then maybe we can then get into some of the questions?
Okay, yeah, well, maybe Sebastian can start off. Okay, I'll start off. No, I just because it was the order of the presentation, but I can.
So I can. So I think I'm
maybe
going to start off.
Yeah. Okay.
Yes, I'm going to start off.
Okay, go ahead.
So I start about talking about thermodynamics and how this sector of physics now help us to understand how our body works. So you know that probably that the human body is governed by millions of chemical reactions, and this creates many big transfer of energies which is exactly what thermodynamics study. And we have to take into consideration, I forgot, that our bodies are a system that needs a balance to basically live. This balance is called homeostasis, which is a balance of absolutely everything that makes our body resilient to outer change. For example, on heat. To understand this idea of heat that I have developed, we have to take an equation that we have to rearrange to express something called the constant of equilibrium, which is expressed in function of the temperature, of the exponential of the free energy change over the temperature and the gas constant. I forgot how it's called.
Right. Just to give a very basic idea of what we mean by, for example, the example of homeostasis is like the human body has to remain at basically the same temperature, regardless of whether outside it's minus 20 degrees or 40 degrees, right? So the idea is that the body has to adapt and manage the thermodynamic inflows and outflows of energy in a way where it maintains always the same. That's what's keeping, this is what the state is in homeostasis. So you're saying that a biophysicist would look at that as almost like a very purely physical problem where you have things coming in, things coming out, energy being the way that we understand how that works would be how to balance all of it.
Yes, exactly. And
go ahead.
You want me to talk about the influence temperature can have, like an example?
Yeah, maybe just a very simple example. No, we don't have to go into very deep details because you did that in your presentation, but yeah, just to help everybody understand.
So talking about the constant of equilibrium, you have this constant defines how a reaction happens compared to something called the quotient of reaction. So usually to have a normal reaction, that quotient is inferior to that constant of equilibrium. But the problem, if we have a shift in temperature, like for example, it gets higher because of how the temperature is in the equation. An increase in temperature will create a decrease in the constant of equilibrium, and if it's this constant ever happens to get under the quotient of reaction, the reaction is totally shifted. So it gets from the products to the reaction, and this can be catastrophic because our body has millions of reactions, and if ever one of them in, for example, a protein synthesization reaction happens to happen wrongly, you won't have that protein anymore. But thankfully, this happens very not much time.
Right, and the formation of protein molecules is what we call denaturing. It's a very, very critical thing, and it's also based on other factors, temperature, but also like the pH, also like presence or absence of certain solvents.
And also heat can denaturate the molecules themselves and have
which is the reason the body has to spend so much energy to maintain this 37 degree temp.
Yes.
You also already mentioned that thermodynamics, we also for detection or diagnosing various problems with something like PETs. And in general, we use excess heat to detect where tumors are. Is that it?
Yes, it's a way to detect a tumor because, as I said before, I can develop what I said. So a tumor cell usually use a reaction called glycolysis, which is not the usual reaction our cells use, which is cellular respiration. The particularity of that reaction is that it produces less ATP molecules, which is basically our energy, but it creates an acidic environment that makes it better for them to expand, which is the whole purpose of a tumor molecule. But because they produce less ATP, they need very much more glucose.
Right.
So therefore, when we talk about the PET scan, if the tumor cells need very much more glucose, the radiopharmaceuticals we injected to the patients will tend to get sent to these tumor molecules, and
they're the ones that need more fuel, basically.
Yes.
Okay.
Yes, and will create therefore more radioactivity and be detected by the scanner.
Okay. So let's maybe move on. I think it was Anastasia. You talked about the role that bioelectromagnet.
Yeah, that's right.
Looking a little bit into that, not necessarily too much detail, but just giving us an overview of what we mean by bioelectromagnetism and how a biophysicist might make use of that or exploit that or
Okay, so bioelectromagnetism, it's a field in biophysics that can be divided in different ways. One classification organizes this field based on two key principles. You have Maxwell's equations and the principle of reciprocity. So Maxwell's equations are a set of four equations that were not discovered by James Clerk Maxwell. He took those equations. There was it was four equations that didn't have much to do one with another, but when you put them together, they were
short idea of what we mean by Maxwell's equations. They're basically equations that explain how the electric field behaves and how the magnetic field behaves.
Yeah, that's right.
First in the vacuum and then inside matter. So that's what the four equations are. They're one of the great achievements of 19th century physics. And in and of themselves, they're just, a very important physics results. But as you're about to say, here's how it gets applied to biophysics.
When you put them all together, you can unify them into one symmetric coherent set of equations. So you've got Gauss's law for magnetism, Faraday's law, and Ampere-Maxwell's law. And so all those four equations help to explain how bioelectric and biomagnetic fields are linked. And they explain that when there are bioelectric fields, there will always be biomagnetic fields and vice versa. So that's why we talk about bioelectromagnetism.
The human body, most living animals in any case, they are what we call like a biomechanical machine. A lot of mechanics, and bones and muscles are joined and something pulling here, but also the activation of all that, which is through the nervous it's a purely electromagnetic, electrical, electrochemical way. That's how we make the link between functions.
Yeah. So there are those fundamental equations, and with that there is the principle of reciprocity, which tells us that the way we measure bioelectric signals, how energy is distributed during electrical stimulation, and how we measure electric impedance, which is resistance, all the things, they follow the same pattern. And so we can all link them in an organized way. And so this principle also applies to bioelectromagnetic and biomagnetic fields.
Can you talk a little bit about what we mean by biomagnetic, or an example?
Yeah, biomagnetic fields, for example, it's in the body. It's the one that, for example, is created by our brain or by our heart. And so do you want to
give us maybe a little bit of details, or at least give us one example of how biophysics helped us make a discovery that wasn't otherwise possible?
Well, yeah, MRI, we talked about it before, but bioelectromagnetism helped us. MRI is a key example of bioelectromagnetism in practice.
Okay. Do you think you can give us a very basic idea of how MRI works?
Yeah, of course. So you take someone and you put it in a MRI. MRI is like a big magnet, we could say. And so you place the patient in this big magnet, and the protons in
areas like this big giant cylinder.
That's right. Yeah.
Empty in the middle. All around that is what we call superconducting magnet,
where the doctor tells you to absolutely not move. Because it can blow the picture. And so your protons with no magnets, they are randomly positioned. But when you put someone in the magnet, all the protons are mostly aligned.
Just again, to make a very specific detail, when we talk about the alignment of protons, we're talking about a spin property of one. So they're aligned when they're exposed to this very powerful field. They all.
That's right. And then there is in the MRI a radio frequency call, and so when a radio frequency current is pulsed through the patients, the protons are stimulated and they spin out of equilibrium, and they are straining out of against the pull of the magnetic field. When the radio frequency field is turned off, the protons go back to their initial position, or all aligned.
Random and in all types of directions.
No, the mostly aligned.
Okay.
Yeah, because you're still in the MRI, and so it comes back to the state where they're all aligned, and the MRI sensors, they are capable to detect the energy released by the protons while they realign with the magnetic field. And so the change of the protons, it depends on the environment and the chemical natures of the molecules. So you won't have the same in your heart, in your lungs, etc.
Some would change more slowly, and that difference,
that's right,
in return to that initial state is what
we're going to see.
Detect the different tissues.
That's right. And then with computers, you can make an image.
Right. Then you have to go through a lot of Fourier transforms and stuff like that, but we're not going to get into that.
That's right.
Okay. Maybe we can go back to Sebastian. You, without necessarily going into specific details.
So nuclear medicine, it consists on the usage of radioactivity to either make a diagnostic of our body. So like I talked about the PET scans. But also what we think the most about is for therapies like radiotherapies.
So important to note that what we mean by nuclear medicine is basically any usage of radioactivity, whether it's natural or artificial, to do one of two things. Either as a diagnostic tool, either as a therapeutic tool that would help deal with or cure certain problems. Okay, so let's talk more about maybe the therapeutic side of it, because you talked about some imaging and detecting technology. What we mean by like radiotherapy.
So for radiotherapies, there are many types of therapies. There are two types. One is the external radiotherapy. I will talk about both, and also internal radiotherapy, which is less known, I think. So basically, what most of us think when we talk about radiotherapy is the big radiation cannon that sends radioactivity, gamma rays, and X-rays to the tumors. So how this works is that the patient is laying down next in front of something we call a linear accelerator. So this works the following way. It shoots very fast electrons towards something called the transducer, which is a very heavy molecule like tungsten, and using a principle called the Bremsstrahlung radiation is basically breaking radiations. The electron kinetic energy is converted into X-rays or gamma rays, and that are shot to where you have the tumor, and you are shot in many angles to ensure that all the rays shoot the tumor in the same direction. And
the idea here would be that how does this help us cure a problem or deal with a problem? What is the basic idea behind it?
So the idea with this is by shooting the tumor, the extreme energy of gamma rays or X-rays slowly destroy the tumor. It can either destroy it totally or partially for after using a chemotherapy or another technique. And the goal is to slowly destroy the tumor.
Okay. So the idea being that we will use the intense energy from something radioactive to direct it in a very specific way towards the specific cells that we want to destroy.
Yes.
Why is this such a powerful sort of therapeutic? Is there a specific aspects of?
Yes, I think I got this because it's not specific for any kind of tumor. It can shoot any tumor. So, for instance, if you have a deep tumor inside your body, like for instance in your prostate, for instance, you can't remove it by surgery, and radiotherapy helps you to destroy it without actually touching it.
Okay, so
And it's usually
what we call non-intrusive because you don't have to cut anything open. You don't have to do operations on the person. And at the
Yes.
same time, it can be very precise, right? You can really precisely decide to what depth you want to go into, to breadth you want to affect the cell. It's very precise, and it has a very high success rate.
I didn't search the success rate,
Okay.
but I guess so. Safe.
You also, just to finish off with this, you mentioned something internal, something about internal therapy.
So yes,
radioactive.
Internal radiotherapies itself separates into two categories. So there is one category that we call unsealed source,
Yeah.
which means that the patient ingests just a radiopharmaceutical. This is we also call it the same way because it's radioactive, but with the purpose to slowly destroy the tumor instead of simply detecting it like a scan. And the other kind is the sealed source. So if there is unsealed, there is sealed, which uses a capsule or a wire
Right.
inserted inside your body that will send the ionizing rays next to the tumor. So instead of sending it all over your body, you have it next to your tumor, but and will also slowly kill it. And
Okay. So the idea is similar, but it's less, I guess, precise in that you're not aiming sort of a radiation gun at the zone, but you're sort of letting it go through your body, and hopefully, I guess that's the chemistry side of it comes into play. Capsule that contains the radioactive gel goes to the right spot.
Exactly.
Okay. All right. So let's move on to one final that I think, Anastasia, you talked about more. Now, this time having to do with fluid mechanics and fluid dynamics and how we use our knowledge of fluids to deal with, again, medical issues or do some sort of...
That's right.
So would you want to maybe give us a short summary of the different aspects?
Yeah, fluid mechanics is a crucial aspect of biophysics because it explains how biological fluids, such as blood or lymph, behave and move and interact within living systems. This field is quite essential for understanding things like circulation, respiration, cellular transport, and medical applications such as drug delivery or hot stents. And so during my presentation, I developed on the key points that need to be determined to study fluid dynamics within a living organism. And so we first had to characterize a fluid. For example, is it stationary, moving, the viscosity of the fluid, which is if a fluid is thick or well,
which it resists
viscous, yeah, how its resistance to flow, and is the fluid Newtonian or non-Newtonian, which is a non-Newtonian fluid is a fluid that would have a viscosity that changes with the applied stress or flow rate. For example, blood is a non-Newtonian fluid because its viscosity decreases as the flow rate increases.
The reason for that would be the presence of not just water as the main constituent, but also particles
That's right.
of cell, platelets, etc.
And is the motion chaotic or non-chaotic, which means is the flow laminar or turbulent. A laminar is just when the motion is smooth and orderly, with parallel layers of fluid moving without mixing. This occurs in small blood vessels most of the time, and is described by Poyer's laws that I'm going to talk about a bit. And you have turbulent flows, which is chaotic with eddies and vortices disturbing the flow pattern. This is seen most of the time in larger arteries during high activity like sports.
So,
or when
sorry to cut you there. A lot of our knowledge of fluid dynamics is applied most.
That's very well, yeah.
Diagnosing if there are problems either.
And not only.
No, go ahead.
There's as well breathe to how we breathe, for example, to lungs. Yeah, that's right. Because at the our lungs, they're divided into many small branches, if I could say. And the smallest part of our lungs is called alveoli, which is like small little empty balls, I could say, or
Like it's like
sacs.
little sacs at the end of the
Yeah, that's right.
the pathways in our lungs, which is where the main exchange of oxygen between.
Oh, that's right. And here, fluid mechanics also applies to the changes, the exchanges of oxygen in this part of the human body.
Okay. Can you maybe just quickly, so we can move on to the question, give us like 2 or 3 applications of our knowledge of fluid mechanics to some like biological systems or
Yeah. For example, well, as we said before, a lot of cardiovascular systems. For example, you have a modeling. So the knowledge of fluid mechanics is helpful to understand how blood flows, how it can work. And so when you know this, you can model how it will interact, and so you can understand conditions like hypertension or arterial stenosis. Arterial stenosis is when a vessel is obstructed by it's obstructed. I couldn't.
Any reason could be a blood clot, could be other. So
yeah, that's right. And so when you know how to model, you can develop treatments, and you can also create medical devices, for example, heart valves and stents, so that you can keep your heart moving, or you can remove the blockage, and prevent it from happening again.
Just as other examples, Doppler diagnostic, where we use Doppler effect, study blood flow in our cell. We are already running quite late. What was it about?
Yes, so it was about new advances in physics, recent trends.
Okay.
And I think I'm only going to talk about one application.
Okay.
Pairing with AI, which is very actual.
Yeah.
So physicians have been able to develop something we call digital twins, which consists of the development of a virtual replica of a physical entity that allows the AI we talked about to simulate physiological behaviors. So, for instance, it can predict how something can react to a medicament without actually putting the medicament to the actual system.
Right.
And the advantage of that, if we ever achieve to develop it at its maximum potential, is that it is patient specific. So the digital twin, for instance, if we're trying to do something with my heart, the digital twin that will be developed using my heart will predict how my heart will react to a drug, but it will be the result will be probably different if it was about your heart or another person's heart. So that's the power.
If I understand correctly, what you're saying is would be using artificial intelligence to sort of model as closely as possible how a person would react to certain medications, so that you don't have to actually do it.
Yes.
And then find some negative results, so you can better tailor the use. I don't know if you guys found anything relating to bioinformatics when you were doing your research?
I don't think so. I don't have myself on
bioinformatics.
Okay. So that's also pretty recent, last 20 years, of using a lot of information technology and computers to model various biological. So we did focus mostly on medical applications. There are other domains which are much more very purely research. Did you guys find anything, for example, about protein folding, like that?
Yeah, but again, it's in medicine, it's for cancer.
Well, that would be the application of it, but the idea being that...
Yeah. It's at Auburn University. I found an article that was saying that scientists were focusing on understanding, with the help of AI, how protein interact with another protein, which is called PDL1. It's a protein just known to help cancer cells evade the detection by the immune system. And so they were consisted on modeling this thanks to AI, model how this protein interacts to understand how cancer can be developed.
Right. So those are, again, much more advanced applications of it going. Okay.
Yeah.
Are you guys ready? Maybe we can move on to a few of the questions from your classmates, and then we'll end our little podcast.
Yeah.
Yes.
Okay. Were there any particular questions that you guys found interesting that you want to at random?
Yes, I think I found a question about how gamma rays can damage the DNA, because we haven't talked about the downsides of
But let's talk about it. Let's talk about
nuclear medicine.
Let's talk about the downsides of nuclear medicine. What do you have?
The downsides are basically the secondary effects of exposing your body to radioactivity.
Which would be?
So we'll again separate this in two categories with tomographies and therapies. So for tomographies, you're probably out of danger because if you take all the precaution, the doses you get of radioactivity are way below the first danger zone. I think it's 100 microsieverts, which make it dangerous if you have a long exposition, which is not the case when you have a tomography. However, for internal radioactive therapies, you have the danger increases because the radio traces can remain and accumulate. So they can have some side effects like fatigue or nausea,
Right.
and create direct damage because of the rays to the tissue next to the zone you apply the radio tracer. Yes.
Right. Go on.
And also, one of the radioactivity can also induce cancer. So if you get exposed too long by trying to destroy cancer, you can provoke a secondary one. But
I'm under the impression that we have made enough advances in recent times that we have a pretty good idea of how to give the right dosage of these things and avoid all these secondary.
Yes, we create the right doses for the patient to be safe.
Right.
And like the question I also saw is how gamma rays itself can damage our DNA and create the cancers and all that can happen.
So let's go into that, because I think that could also touch on the general idea of why radioactive is the danger. What is the danger? Why is it something so much fear?
Well, radioactivity is when a very big and unstable nucleus of an atom spontaneously disintegrates and sends various particles to various particles in its environment. And there are many kinds of particles, which like the alpha rays, which are helium nucleus.
Yep.
Beta rays, where positons or electrons are emitted, or the ones I have talked about, the gamma rays, which are purely photons,
Right.
but very energetic
High energy
photons.
electromagnetic radiation, the highest energy of the three types of particles,
Yes.
and the most dangerous.
So gamma ray can be dangerous because it can firstly create direct damage. So that's what we mostly think about, because they act, the ray acts against our DNA. They act as a sword, cutting the strands. They can either cause single breaks that can be easily repaired by our molecular proteins, but the worst are the double strand breaks, which can cause repair errors and lead to mutations. A bad mutation can itself lead to a cancer.
Right. Or
But
be lethal.
yes. But one I also discovered by trying to answer this question is indirect damage. That is 70% of the radiation damage induced in the DNA. So basically, when a gamma rays can be absorbed by our water molecules next to the DNA,
Yeah.
and they create something we call free radicals.
Yeah.
So these molecules lack an electron, and therefore are highly reactive for them to seek another electron, which they find usually in the bases of our DNA, which can cause themselves strand breaks, and it's very
Oxygen
dangerous.
being the main one of these free radicals, which is very, very problematic. Okay. Maybe we can go to one or two other questions. At the very beginning, you mentioned how biophysics, one of the main first breakthroughs of discoveries was DNA double helix structure DNA. Maybe Anastasia or Sebastian, whichever. Do you want to give us a very, very brief?
Yeah. From what I read, X-ray crystallography and nuclear magnetic resonance spectroscopy. There are techniques in biophysics for elucidating the structures of biological macromolecules, I think.
Do you remember?
X-ray.
You might have done a little bit of it in physics this year in Terminal. How we can use waves basically to figure out the distances about.
Yeah. No, I don't quite remember.
So maybe you haven't done it yet, but I know in my class we just talked about.
Oh well, we started it. Just.
Right. So basically, that's I think the really simplified things that would through a network, a structure which has some sort of regular spaces and obstacles produced, and then the way diffraction pattern comes out the other side, you can know about the distances, the different parts. Maybe we can go for one final question. Did you guys have another question? Did you guys find anything about? So, it was one of your classmates talked about the role of quantum mechanics.
Yes. Again, quantum. The researchers they use quantum mechanics and methods to study biomolecular structures and reactions, improving computational modeling of biological systems. This method help explain protein folding, ligand interactions, and electron transfer. Ligand, I'm not sure if everybody knows ligands. It's like a molecule that can link to another one, I believe.
Yeah.
To a macromolecule, and it has many functions like catalysis or act as like an enzyme or transmits signals and so yeah, most of the time quantum mechanics are used to explain how things work.
Right. So what you're saying is that basically quantum mechanics plays a role in the understanding a lot of other phenomena. What has more of an application?
Well, from the article that I read, it was like it kind of acted just like biophysics. You take quantum phenomenon and you apply them to biological systems, just like with physics.
So, if you guys are okay with it, I think maybe we can stop there. Maybe I'll ask you one final. Personal. What did you guys think of the topic? Was it something that you guys had any knowledge of before? Was it something you discovered? What did you find interesting, easy, difficult about it?
For myself,
Go ahead, Sebastian.
For myself, I got this topic because I found it interesting, but I didn't have many knowledge of this.
It's not something that you would see in high school because you're just learning the physics part of it. So to already be applying it to other things, I think a bit too early.
So I also chose this topic because I knew it will talk about nuclear medicine, and this particularly interested me. So like that's the biggest part I talked in my presentation also. But I also found the thermodynamics very interesting. So it made it fun to research on the topic.
Okay, and Anastasia?
I was a huge fan of the topic, I think, because I knew more about the medical part than the physics part, and
because your plan
next
year are to continue towards medicine.
Yeah, medical school. So most of the time I knew how things worked, but I didn't know why. And understanding why it worked that way, and be able to understand it with concrete like laws, and it works like this because physics laws are applied. I find it particularly amazing, and I learned a lot of new stuff about, especially recent studies about protein folding, AI, and as well quantum mechanics and nanoparticles, for example, for drug delivery. And I was really happy to learn about that for the presentation and for the podcast.
All right. So I think we're at a good place. We're almost at an hour, which is much longer than I had imagined. But you guys, in your presentation, that no way of cutting it short. So thank you to both of you, Sebastian and Anastasia, for working it. I hope you learned something from it. We certainly learned a lot from it.
Thank you.
Okay,
Thanks.
so thank you very much, and don't forget.
Bye.
Bye.
So, that is Physics Bites for this week. We hope that you enjoyed this episode, and we would love to hear what you thought about the topic in the comments section. If you did enjoy the show, then subscribe to our podcast, share it with your friends, and be sure to leave us a 5-star rating on whatever platform you're listening on. And more important than all of that, come back soon for a new episode.